So we mentioned something about normal flora bacteria. We said that not every bacteria can cause a disease. So let's talk about that idea in more detailed manner. First of all, of course, we need to say that it is really so that not every microbe can cause disease in human. We have natural immunity against some uh, germs and microorganism that is able to produce a disease in medical practices known as a pathogen. Pathogen means any microbe which can cause disease in human being. And that pathogen have an ability to cause a disease which is known as pathogenicity. So pathogen, it is a quality of microorganism, and that quality can be evaluated by pathogenicity, by ability of one organism to cause a disease in another organism. And organism which suffers from pathogen known as a host. And of course, in human medicine, man is a host, and bacteria or some viruses are pathogens. And pathogens are classified into two uh, groups, obligate pathogens and potential pathogens. Obligate pathogens, it is microorganisms which do not associate with the host without causing a disease. Means that any contact with that organism will obligate lead to disease. Or if we are, if we are immune to that organism, of course, disease will not be possible, but microorganism will not be able to fix in our body, on the surfaces of our body. It will be eliminated. Potential pathogen, it is microorganism which lives as a commensal or parasite in our body or on the surface of our body without producing disease until weakened in hosts, anatomical barriers, or decrease in resistance and immunity will occur. So it means that in common life, while we are strong, we have good nutrition, we have no neuro stresses, we are relaxed and fine. Potential pathogen cannot cause a disease in us. But later we will discuss those reasons. If something will happen, that microorganism can transform to pathogenic and cause disease in us. And mainly, potential pathogens are presented by normal flora bacteria, which is microorganisms which are frequently found on or within human body of a healthy person. Means that those microbes should be found almost in everyone, and that everyone should be healthy. And those microbes are usually living as a commensal flora or parasite flora, but we're not suffering from them. And of course, that idea of normal flora is known already for a century, and uh, we know that normal flora is obligate part of our life, of our system, and of course, that flora have multiple benefits. For example, normal flora occupying all suitable locuses of our body, which are very good for nutrition, they're suitable for temperature, they're hided of sunlight, and if pathogenic microbe will try to invade, it needs some place to fix and to feed, but those places are already occupied by normal flora. And moreover, normal flora during its life cycle can release multiple substances which are harmful for pathogenic bacteria. They can produce bacteriocides, they can release some acids, alkalines, and all accumulation of those waste products will be not suitable for pathogenic bacteria. It became more difficult for pathogens to infect our bodies. So it is confirmed and proved by multiple studies that normal flora is responsible for rising of overall immune state. Means that our tissues are heavily inoculated with normal flora bacteria, millions of bacteria living in us. If we will measure them, kilograms of bacteria are living in us. And of course, constant presence of microorganisms in our tissues will induce immune response because more number of cells, macrophages and other cells are required to monitor that border where bacteria is present and where our tissues are present. And of course, multiple accumulation of immune cells known as lymph nodes or lymphoid tissue are formed due to constant presence of that bacteria. And our immune system is always um, activated because multiple microbes are there. And maybe they are normal, but how can we know that they are normal if we will not study them? That's why our cells need to monitor state of that bacteria, monitor our tissues, and a lot of cells are attracted to the sites where normal flora is present. And that's why we have very good developed lymphoid tissue. Oral cavity, if you will take that Pirogov ring, a lot of lymphoid tissue accumulation because we constantly filled in with microbes here. Intestine, pairs, patches, huge accumulation of lymphoid tissue because intestine is rich with bacteria. And of course, during life cycle, some bacteria are dying, some are appearing new, and those dying bacteria can release toxic products which are toxic for us, endotoxins, for example. The same E. coli can be accidentally absorbed into blood vessels and get into the blood and endotoxin is produced, and that endotoxin is stimulating activity of overall immunity as well. And it is was shown in newborn kids that kids who was growing without normal flora they have much weaker immunity because it is not stimulated by normal flora. And of course, one of the most known functions it is a digestion function when microorganisms present in our intestine, in our bowel, they can digest some heavy nutrients which cannot be digested by our organism. They help in digestion of some plant food and other. And of course, some bacteria, E. coli included, can produce essential elements of vitamins. E. coli can produce vitamin K, for example. It is not synthesized in our body and it is present in low amount in food, but E. coli, which is normally living in our intestine, can produce that product. It is a waste product for bacteria, but it is very suitable element for us. And bacteria excreting that vitamin outside because it's not required for bacteria, but it is absorbed in our bloodstream and used by our cells. So of course that function is extremely important. And again, it was confirmed that even in oral cavity, some microorganisms can start already digestion and proper digestion starts in mouth. And without mouth digestion, some components cannot be utilized in intestine and mouth flora is very important as well. But as everything in our planet, we have two sides of medal and we have some penalties as well. And the main penalty that bacteria belong to normal flora can cause disease. As we said, by certain circumstances, bacteria can become pathogenic for us, and it is very important penalty. Again, presence of multiple species of bacteria in different organs and tissues can make confusions in diagnosis of diseases. Because if normally we would not have any bacteria, then patient suffers from some infectious disease, you need to take sample, and that sample will always contain only pathogenic bacteria. Diagnosis is easy. But normally we all are rich with bacteria. And when you will take a sample from skin, from nose, from oral cavity, from intestine, any area you will take, you always have mixture of microbes. And of course, it is extremely difficult to understand which one is normal flora, which one is pathogenic, and how to suggest that maybe it is normal flora became pathogenic, or it is still normal flora. And of course, diagnosis became more difficult because of that bacteria here. And one more problem, it is a modern problem, it is a genetic problem. Our normal flora bacteria can release plasmids, for example, which coding for antimicrobial resistance. And that plasmid can be grabbed, can be absorbed not only by normal flora, it can be acquired by pathogenic bacteria. And of course, pathogenic bacteria can become more dangerous to us due to our normal flora. And there is one more reason why we try and maximally avoid intake of antibiotics. And we trying to keep the drug as a reserve, not to give it to everyone. Because maybe you will treat some simple infection faster, but your normal flora, which will remain here for all your life, it will acquire new property resistance to your used by you drug. And that resistance is mainly coded by or can be coded by such genetic elements as the plasmids. And your flora will start secretion of plasmids in environment, and sometimes it can share with new pathogenic bacteria, and one antibiotic will be lost, or even one class of antibiotic can be lost for your therapy options. That's why, again, it is important to use those drugs only when it is essential and required by some obvious circumstances. But in simple infections, try to avoid those drugs because your flora will remember which drug you used and how to resist that drug. And new pathogenic bacteria can acquire that property as well. So we need to say that, of course, not every part of our body is rich with microorganisms. We have locuses which are normally sterile and have absolutely should have no bacteria and no microorganisms at all. Of course, it is first of all blood and CSF. All internal liquids should be absolutely sterile, and it is kind of normal phenomenon. Well, transient bacteremia can occur due to different reasons. Some aggressive tooth brushing or from intestine, some microbes can be absorbed. But normally, blood should contain no microorganisms. Of course, cerebrospinal fluid or CSF or liquor, it is protected with managed encephalic barrier, and no microbes should present, and liquor as well should be absolutely sterile. All internal organs with no contact to the environment means if organ have no open access to environment, liver, spleen, heart, any you will mention, it should be absolutely sterile because bacteria have no opportunity to get into that organ without invasion, and normal flora should not invade. We have very good protection in respiratory tract. We have the Pirogov ring, a lot of ciliary epithelium, and actually respiratory sinuses, middle ear, and lungs, starting from trachea. They should be sterile as well. Again, it is possible to have some bacteria for short time period, but normally no flora should present here. And in women, cervix and uterus should be sterile. Bladder should be urinary bladder should be sterile in everyone, and even urine normally should be sterile. Again, we have that phenomenon when flora from skin can slightly pass to uh, first part of the to lateral part of the urinary channel in men, and it is and in women even as well. And it is possible to have some microbes in the first portion of urine. That's why it is always obligate to collect urine in precise manner. First portion should be dropped out in the sewage and toilet, and only second portion should be collected for further studying because first portion will wash out all microbes which stick in the skin, and that flora is not from urine, it is a skin flora. And when that flora will be removed, second portion of urine should be absolutely sterile. And if you talk about organs and tissues which are contaminated with microorganisms, all microorganisms or all flora is divided into two main groups. First group is a resident flora. It is a group of microorganisms which are constantly living in our tissues and our bodies. They are always here. Usually they are presented by very few species, but they are always found here in every healthy man. And microorganisms that usually occupy a particular body site are called resident flora. And cells of resident flora outnumber a person's own cells 10 to 1. It means that every one single cell of our body is covered with 10 bacteria. And of course, it is very huge number because actually bacteria uh, present in higher amount in our bodies than cells we have in the same organ. And transient flora is some microorganisms which can be accidentally found in some locus of our organism, and they colonize people for hours to weeks, but they do not establish themselves permanently. It is a resident flora. They can be found here, but if you will perform some repeated samples, you will not find that microorganism in constant manner here. They are present for a few hours, maybe days, but then they should be removed and should not be here. Usually that bacteria can go here from neighborhood organs. For example, the same bacteria from skin can invade into urinary channel. Bacteria from nose, for example, can get into the skin as well, but it is accidental finding and very fast it will be removed and replaced. So as well, we should remember that even resident flora can differ in different geographic regions according to climate, according to nutrient behavior. Flora can be different even in the same age people, the same gender people, but according to for example, race flora can be a little different. Of course, with the age, flora is differs because newborn kids are delivered sterile and they have no bacteria at all. In gender, of course, sexual organs especially have very different microflora, and hormone status is different than men and women, and flora can a little differs. But still, we have some common microorganisms which should be identified in every person, whatever race is or geographic region. So microflora of different organs and tissues, of course, presented by different microorganisms. Some can be identified in several locuses, but we have kind of common rule that skin and external organs are for gram-positives and mainly cocci. And internal organs, intestine and some stomach is for gram-negatives. That common rule it is usually working. So the main representative of skin flora is Staphylococcus epidermidis. It is even named epidermidis because it is widely found in skin. And that bacteria is found in everyone. It is living here constantly and not causing any problems. We can have Staphylococcus aureus. Again, it is fine to have it on skin because usually it is our own flora and it is not dangerous. But again, that species of Staphylococcus aureus is not homogeneous, and we have some strains which are pathogenic. And of course, those pathogenic strains, they should not be identified in the skin as normal flora. Only potential pathogens should be identified in the skin. And one more bacteria, which is actually very known for almost everyone, it is Propionibacterium acnes. Name is given because of unique lesions formed mainly in the skin or face. Disease is known as acne. It is formation of black dots when channels or pores of our glands are obstructed by bacteria. It will accumulate by secretion of our glands can obstruct channels of pores, and bacteria can grow here and produce those kind of inflammatory elements on the skin. Usually it is juvenile problem, and with the age it will be resolved when hormone status will be changed. And actually, only those bacteria can be found in skin as a resident flora. The rest bacteria should not be put it here. And very common mistake, and I know that it is even written in Panicker book, that you can find widely E. coli on the skin. You can find some uh, other gram-negative bacteria on the skin. It's not really so. It is possible to have E. coli as a transient flora, for example, on the skin of perineum, because bacteria from intestine can accidentally grab to the surface of the skin, and of course it can be found here. It is possible to have E. coli in hands if you forgot to wash your hands after the toilet. It is again possible, but E. coli is not normal flora of the skin. It is normal flora of intestine. That's why it is even named coli, means colon, means intestine. And it's not flora of the skin. It should not be here. Yes, it is possible to have it for some short time period, but it's not normal flora here. It is transient flora. It's not normal. So, uh, if you talk about conjunctiva, it is again kind of epithelial tissue. We can have the same bacteria here. One unique microbe which is present only in conjunctiva is Corynebacterium xerosis and few species of Moraxella. But again, mainly any bacteria on conjunctiva is transient bacteria because we have very good production of tears. They contain multiple salts and biological substances to suppress flora. We always blinking to remove bacteria, and actually, in normal way, conjunctiva should be sterile. But very often and frequently, we have those listed microorganisms in conjunctiva. It is fine. It's not meaning that it is some pathogenic bacteria. They can be identified here. Again, they can grab here from skin, or they can present here for multiple times because it is just most suitable tissue for their leaf, but still it is normal bacteria. Other bacteria should not be found here. Usually they have some pathogenic properties. So nose, of course, have very similar flora to skin because actually it is almost the same locus, and some virulent streptococcus can be identified here and few non-pathogenic species of diphtheroides, but usually flora is approximately equal. And the main organ, respiratory organ, the most important and the most dangerous is the throat. Again, we can have Staphylococcus epidermidis here, and we have Streptococcus viridans as GIT flora already here. Staphylococcus epidermidis is normal bacteria of the skin. It can again present here because it is the same epithelial tissue. Viridans Streptococcus is very wide group of digestive flora, and they perform a lot of multiple suitable functions here. It is possible to have transient flora of Streptococcus pyogenes if it is non-pathogenic, if it's not causing a disease. But usually Streptococcus pyogenes is identified as a carrier of the disease. The same is for Streptococcus pneumonia, for Neisseria meningitis, and for Haemophilus influenza. About 15% of population can have that bacteria in normal flora. It is fine if that bacteria is not causing any disease, but some people don't have it. And moreover, those bacteria fall as bacteria. Under some circumstances, they can cause diseases and very serious diseases. Streptococcus pyogenes is responsible for scarlet fever and different types of tonsillitis. Streptococcus pneumonia can cause pneumonia and meningitis in kids. Neisseria meningitis can cause meningococcemia and meningitis as well. And Haemophilus influenza is named influenza because usually it is joined with influenza virus, and it can cause bacterial super or co-infection with influenza virus. So if you have that bacteria in some subject without any symptoms, it can be fine. But again, that bacteria, you should remember that that bacteria can be very dangerous. And if decrease of immunity will occur, then that bacteria can cause serious diseases in people. So if we will talk about genital urinary tract, of course, flora is different in men and women, but external genital urinary tract in men especially is usually presented by Mycobacterium smegmatis. It is caused, have the name smegmatis because It is found in genital tract. Uh, in women, it can be presented by Peptostreptococcus, by Bacteroides. Uh, Usually, that Bacteroides is a flora of GIT, but it can, because of close anatomical location, it can contaminate genital urinary tract as well. And some viridans streptococcus can be identified here as well. In urethra, we can have the same flora as in the skin. It is some Diphtheroides species, non-pathogenic Carinobacterium, Staphylococcus epidermidis, and even some species of Mycoplasma can be identified here. But again, we need to remember that Mycoplasma can can present pathogenic bacteria, and you need to evaluate pathogenic potential of microorganisms. So in vagina, we can have multiple lactobacillus, mainly acidophilus, but lactobacillus and streptococcus, multiple species of streptococcus can be present here, and they perform protective function. They secrete a lot of substances for protection of pathogenic bacteria. We can identify Peptostreptococcus anaerobic bacteria here, and some Diphtheroides as well from skin can appear in genital urinary tract of women. And again, we need to remember that vaginal flora is differs according to age. In young and kids, flora will be absolutely different. When sexual activity is established, flora will be changed significantly. But in adults, usually vaginal flora is presented by those bacteria. Of course, the main organ, the main system even rich with bacteria, it is digestive tract. And the most contaminated place of our body is oral cavity. It is presented by more than 400 species of different microbes, actinomycetes, and other. Of course, we can have a lot of anaerobic bacteria here. We have multiple invagination, cavities, and a lot of bacteria can be identified here, such as Fusobacterium, Bacteroides, even some fungus can be identified here, actinomycetes, and multiple species of Streptococcus. And flora of oral cavity can be different according to locus of oral flora. In teeth, flora can be slightly different than in the whole oral cavity. And we have two important species, Streptococcus mutans and Streptococcus sanguis, which are responsible for formation of dental plaque in the case of poor hygiene of oral cavity. And again, that bacteria is fine until you keep it under control. If you will forget to clean your teeth regularly, then that bacteria can lead to accumulation in higher amount than it should be here. It will release a lot of acids and aggressive substances. They will cause a defect of our external structures, our emailium, and they will lead to formation of dental plaque. After that, concomitant bacteria can join and cause serious destruction of our teeth and our tissues. So esophagus and stomach can be even sterile, because esophagus is the place where alkaline media of oral cavity is mixed with acid stomach juice, and pH of esophagus is always not stable. It is always changing from alkaline to acid, from acid to alkaline, according to uh, our rhyme and food intake. And only lactobacillus can be adopted to that condition and can be found here, but usually esophagus is free of bacteria. It can be sterile, it is fine. Lactobacillus can be identified, it's not a problem. And in stomach, the same very aggressive acid condition allows to survive only for lactobacillus and maybe Helicobacter pylori. That last microorganism, H. pylori, is a very questionable microbe. We will discuss it in next semester when we will talk about anaerobic bacteria. So, according to modern data, according to 2020, WHO performed some investigation, and it is already identified in 75% of worldwide population. Means that three of four persons are infected with that microorganism. So the same manner, until that bacteria not causing any problems, it can be evaluated as normal flora bacteria. but frequently that microbe can act as the reason of stomach cancer, stomach ulcers, and even peptic ulcers. So very unclear microorganism, it can be dangerous and lead to serious consequences. But a lot of population already infected with that microorganism, they don't have any problems, any doubts, so that microorganisms can be evaluated as normal flora. So, if you talk about flora of intestine, it is again different: small intestine and large intestine. You can see that uh, in small bowel, we have mainly coliform bacteria. It is representative of family Enterobacteria, Enterobacteriaceae. Of course, it is E. coli first of all, and a lot of E. coli is identified in small bowel. We can as well identify Lactobacillus, a lot of multiple species of Streptococcus, and in the lowest part of small intestine already we can identify some bacteroid species, but mainly small bowel is filled in with facultative anaerobic bacteria. Large bowel is the distant place of our GIT. It is presented by multiple facultative anaerobic and obligate anaerobic bacteria. The main digestive flora of large bowel is Bacteroides and Fusobacterium. As well, we still have Lactobacillus, we have some Enterobacterium and other bacteria. So we have the idea of dysbacteriosis. It is the state when quality or quantity of normal flora bacteria can be changed. So we can find a lot of different definitions of dysbacteriosis. The one we like most of all is dysbacteriosis is qualitative and quantitative changes in the composition of normal flora, means that it is not obligate to have some pathogenic bacteria invaded in intestine. Even this balance of normal flora can lead to dysbacteriosis. Of course, usually we refers to intestine because mainly bacteria is living here in large number. It can be some vaginal dysbacteriosis or oral cavity dysbacteriosis, but rarely. And of course, usually dysbacteriosis of intestine will manifest as discomfort in abdomen, gas production, pain, tenderness, maybe diarrhea, nausea, or contrary constipation and bloat in abdomen. But uh, different symptoms can appear here. Of course, if it is some vaginal dysbacteriosis, it can manifest with unpleasant odor, with some discharge of white color, especially if it is fungi, and by discomfort and burning sensation in corresponding organ. And the main question is why it is happening, why these bacteriosis can appear in human. We have multiple reasons, but they all can be combined into five main groups. First of all, it is concomitant disease or decrease of immunity. When natural protection decreasing, potential pathogens became stronger and they can easily invade in our tissues. It can be some local trauma. It is skin trauma, scratches, or trauma due to different medical procedures, invasive procedures, some endoscopy or surgery. Of course, trauma of skin will open gates for bacteria, can easily invade deeper in the tissues and cause some pleural inflammation. It can get into blood and spread over the body through the blood. And of course, any type of trauma in internal organs or external surfaces can lead to dysbacteriosis, and normal flora can be transformed to pathogenic bacteria. Sometimes change of diet, very serious and rapid change of diet can lead to dysbacteriosis as well, because our bacteria is adapted to the type of food we are using daily. We have unique uh, composition of bacteria, and we have even the definition of fingerprint of flora. It is possible to identify human by unique composition of bacteria in intestine. It is one of the things used in by criminalists in identification of the person. And of course, if you will change the diet, composition of your bacteria can be changed, and the change will be manifested with some discomfort symptoms. And sometimes traveling or change of climate can lead to dysbacteriosis because again, new type of food, new bacteria which is naturally identified in that geographic region, change of temperature can lead to different type of stresses and bacteria can be changed as well. And of course, the main reason and the most important and difficult reason, and it is one of the uh, reasons doctors are responsible for, it is different type of aggressive treatment, massive antibiotic, antimicrobial treatment. It is immunosuppressive treatments during treatment of cancers, so autoimmune disorders, and different drugs which can suppress our flora, kill our bacteria, or suppress immunity, suppress our immune cells. Of course, they will lead to disbalance of flora, and any of that reasons, or especially if you will have combination of reasons, you travel somewhere, changed diet, and you took some antibiotics and you was injured, and of course, this bacteriosis is one of the very first things you will have. So, what can we do to prevent or to improve state when this bacteriosis already developed? First of all, of course, we need to improve our food behavior and we need to have good feeding. If it is possible, we should raise immunity. Some common therapy, vitamin therapy, or some food supplements should be used to enrich immune overall immune state. Of course, we should have rational chemotherapy. Means you should prescribe drugs, any type of drug, only by certain indications in correct manner, in correct dose, in correct frequency, duration, and etc. And we have several groups of drugs which can be used as for treatment and for prevention as well. First of all, it is probiotic. Probiotic or probiotics, it is a complex preparation of microorganisms composed normal flora. Means that probiotic contain microbes which are naturally already present in our body. But of course, if you know that some microorganism is absent, it is present in low amount, it was suppressed by some drugs, you can give them as a separate drug, and the drug will restore amount of your bacteria. So it is drug contain one of few known species of normal flora bacteria. We have prebiotic. Pre means something preceding bios life. It is a group of drugs containing compounds which cannot be absorbed in the bowel, but they will provide favorable condition for stimulating growth of normal flora. Means it is different combinations of vitamins, amino acids, some food supplements, growth factors, which will stimulate the growth of flora which is still alive in your body. And a newly group of drugs, symbiotic. Syn means synergy. It is a combination of probiotic and prebiotic. It is a compound which contains both alive microorganisms and some food supplements for your bacteria. And one of the last products which are again available in pharmacy nowadays, it is eubiotic. Eubiotics are non-pathogenic microorganisms which are not the part of normal flora, but they can restore normal flora and they can inhibit or suppress harmful pathogenic bacteria. Means that it is some intermediate microbe which is not required for us, but it can protect our bacteria and provide suitable conditions. Maybe they will release some substances to support the growth of our microbes. They can help to resist some pathogenic bacteria and resist some infections. And again, they will improve overall state of intestinal health. So nowadays, of course, it is one of the most popular food products. If you will go to some grocery store, you will identify, you will find a lot of different yogurts, milks, and a lot of food supplements enriched with microorganisms. Lactobacillus, some bifidobacillus, a lot of different bacteria can be added into the food. And it is one of the common measures to support normal state of our normal flora and support health of our intestine. Of course, it is fine to use them. They are not dangerous. They will support your microorganisms, and we have even separate kind of branch of food industry. It is yogurt therapy and some healthy food. A lot of different products are available, and of course, we can easily buy them and use it is fine. So one more thing which is poorly described in your books and you're usually difficult to answer the question. It is gnottobiology. So gnottobiology, it is study of animals growing either free of microorganisms or associated only with specified microorganisms. Usually animals are grown in sterile environment. And initially, all animals delivered here, they are fed with sterile food, they are provided with sterile water, and absolutely no bacteria is present in those animals. So currently, that science gnottobiology is directed towards studying the reactions of germ-free animals after they have been inoculated with specific known microorganisms. Uh, so the main task of gnottobiology is to take sterile animal infected with one only known species of bacteria, usually belong to normal flora, and take a look what difference will appear between absolutely sterile animal and that animal inoculated with bacteria, or what will happen with animal after inoculation of bacteria. Again, the main task is to study why do we need normal flora, which benefits it's providing for us, which penalties. And we cannot study it in healthy humans because we all have hundreds of different bacteria. And of course, we cannot know which exact properties associated to precise bacteria. But if you take sterile animal and inoculate only with one microbe, it will be obvious that if some property appeared, that property is related to microorganism. If something became worse, then that worsening is due to that precise microorganism. And moreover, sterile animals can be used to study pure immune response. Means that if subject have having normal flora will be infected with pathogenic microbe, our flora will interfere with that pathogen. And of course, we cannot study pure immune response. But if you take absolutely healthy animal and inoculate with pathogen, you will see, you will study pure immune response. And germ-free animals are widely used in different medical and microbiological studies to evaluate a role of different microbes, pathogenic or non-pathogenic in our life. And those chambers, as provided on the slide, are used to grow animals there absolutely isolated from environment. Only filtrated air is provided here, sterile water, sterile food products, and those sleeves are used to have a contact with an animal. So no contact with environment is present in those animals, and all procedures can be done only in inside the chamber. And those animals are sterile. We can grow any type of animal according to the size. Chamber can be bigger. Usually it is mice, but we can grow guinea pig and some other even dogs and cats. So the main task of gnottobiology, again, let's list it by points. First, it is explores the effects of microorganisms in physiological diseases. Second, it explores the role of bacteria and protozoan in protozoan and viral infections. Third, it explores contribution of bacteria to nutrition, immunity, and physiology. And of course, gnottobiology identifies specific causative agents in infectious diseases. And it was initially part of microbiology, but now it is growing into separate branch of science. Gnottobiology is a separate science. Of course, it's very difficult and specific, and we have separated laboratories who deal with sterile animals and performing multiple experiments. So for current lecture, that was the last slide. We will finish for today. Save your knowledge. Bye bye.